Alright, so I'm going to be doing my presentation on Pseudomonas aeruginosa and chronic lung infections. Um, Pseudomonas aeruginosa go through evolutionary change and adaptation during chronic infections of cystic fibrosis. Like less virulence factors transition to a biofilm-associated lifestyle and increase antibiotic resistance. They show a lot of phenotypic diversity, like antibiotic resistance and toxin production, making it hard to diagnose and treat. P. aeruginosa infection in CF patients is pretty horrible, and it's a genetic disease that has a defect in a transport protein, causing there to be sticky mucus, most notably in a respiratory tract. CF patients are susceptible to chronic lung infections, and the main cause of the morbidity and mortality associated with the disease. And the most common pathogen is P. aeruginosa, which is a bacterium that can cause a wide range of infections. And it's flexible. It has a large genome, which has a lot of regulatory genes, as well as genes involved in the catabolism, transport, and efflux of organic compounds. In CF, we figured out that if we initiate forceful early therapies where treatment starts as soon as the pathogen is detected, it helps to delay the onset of the chronic infection. However, once a chronic infection is recognized by P. aeruginosa, the studies claim that it is nearly impossible to destroy. During the chronic infections, CF patients produce mainly sputum samples that are used for analysis for diagnostic purposes, to help us identify the pathogens and antimicrobial susceptibility testing. So, as far as phenotypic adaptations that are commonly associated with CF infections, the CF lung is a pretty stressful environment for invading bacteria. The P. aeruginosa populations have to overcome these challenges to survive. The stressors are osmotic stress due to viscous mucus, oxidative and nitrosedative stresses due to host responses, sublethal concentrations of antibiotics, and other microorganisms. P. aeruginosa goes through changes in response to these forces during the chronic infection. Analysis of isolates showed that the rise of mucoid colonies caused by overproduction of the polysaccharide alginate, which is considered to be a marker for the transition to chronic infection. Alginate is one of the three exopolysaccharides, along with PEL and PSL, that play vital roles in the creation and preservation of a biofilm matrix that can offer P. aeruginosa protection from antibiotics and host responses. Other adaptations are accumulation of oxotrophic mutations in the amino acid-rich lung environment, loss of motility, and the emergence of hypermutators, which show more mutation rates due to defects in the DNA repair mechanisms. CF patients often have long and intense therapy with antibiotics, so developing antibiotic resistance is a common adaptation. Another repetitive thing that you would see in CF isolates is for P. aeruginosa to become defective. When it comes to its key virulence factors, like type 3 secretion and QS system or quorum sensing system, gathering of the mutations in the gene encoding the key QS regulator, LASR, causing loss of QS regulation is pretty common. Other mutations is often associated with CF isolates of P. aeruginosa include mutations in the GACS and the RED-S, genes implicated in the switch acute and chronic virulence. In the accumulation of virulence factors, mutation appears as a P. aeruginosa is adapting to lose its acute virulence due to chronic infections, but many virulence factors can still be detected in patient sputum samples during chronic infections. As far as P. aeruginosa virulence factors, it's typically associated with acute infections, so the expression of virulence genes is controlled by regulatory circuits and multiple signaling systems. It makes a group of secreted virulence factors utilizing its type 3 secretion system to secrete different exotoxins, like XOS, U, T, and Y, and quorum sensing systems to control many of the virulence factors. And the secretions of toxins coupled with phenotypes is important for acute infections, and it's been said that the GAC system network controls that reversible transition from acute to chronic infections. That's based on a switch between acute virulence factors and chronic virulence factors, like a biofilm formation. As far as genomic analysis of adaptation using sequential isolates, there's been a lot of studies comparing clonally related longitudinal isolates, mainly contrasting the mutated genes and isolates from early and late in the infection process, and it showed common mutations falling into different functional categories like virulence, motility, transport, antibiotic resistance, iron acquisition, DNA replication or repair, transcription, translation, cell division, or metabolism. And mutations in genes encoding key global regulators are common, and the traits affected by these mutations are called patho-adaptive traits. An analysis was done, and a lot of which showed a transmissible lineage, DK2, and after initial transmission, sublineages evolved independently in patients, storing patho-adaptive mutations. It was then shown that hypermutator lineages can coexist with non-hypermutators, creating evolutionary pathways, and all the studies done show evidence for adaptive evolution, with selection for mutations that are helpful in the CF lung. For instance, there's studies for adaptation towards iron acquisition from hemoglobin repeatedly and independently across multiple patients, and Aeruginosa has regulatory and metabolic networks in the lung that modify gene expression levels and alter the metabolic fluxes. Mutations that happen earlier on in the infection are in global regulatory network control hubs, with other mutations occurring later and leading to fine-tuning. So then we ask the question, what are the drivers of the pathoadaptation? So the evolution of certain traits or genes freely in different patients suggests positive selection, leading to recognition of the set of pathoadaptive traits. And there's new phenotypic and genetic descriptions of how natural selection goes after P. aeruginosa populations in the CF lung. But I couldn't really find a lot of the information on why these traits and genes go through selection. And it's probably because the CF host environment is really complex, so we don't truly understand it completely. The only thing I can really think of is where the selective force can be linked to the evolutionary responses for antibiotic resistance evolution. There was one study that I looked into that P. aeruginosa adapting to the CF lung, and I was researching the fitness of particular alleles of the penicillin binding protein 3 and how it could be linked to the use of particular antibiotics. I found another article that did a study using a nematode host, and it showed no effect of host immunity on P. aeruginosa adaptation. Virulence traits were lost in the, with and without immune selection. Similarly, adaptation to the CF-like conditions of artificial sputum medium selects for mutations that cause a switch to a immotile biofilm lifestyle, suggesting that the sputum environment itself is sufficient to select against motility. Moreover, growth of P. aeruginosa and flow cells selects for mutations causing mucoidy and loss of pilus-dependent motility, suggesting that simply dwelling in a biofilm is sufficient to cause evolution of these characteristic CF-associated phenotypes. Other common adaptations include changes in metabolism, DNA repair, iron acquisition. However, there's a clear need for careful experiments testing evolutionary hypotheses about the drivers of selection within the host of which entangle the complex, multifaceted environment. Outlines our current knowledge with respect to adaptations driven by social interactions, demonstrating how experimental evolution can help us both generate and test hypotheses. As far as adaptation and social evolution, the transition from acute to chronic infection can radically alter costs and benefits associated with the secretion of extracellular metabolites. Some metabolites are costly to produce when not needed, which may explain the redundancy of virulence-associated genes observed during CF infections. However, secreted metabolites are often costly to produce, so when they are crucial for growth, their loss might be selectively advantageous. Providing there remain enough producing individuals to compensate for the small reduction in metabolite concentrations, hence the non-producers that can still reap the benefits of metabolites produced by others while paying little or no cost. So for instance, individuals that produce costly iron scavenging cytopore pyridine can be exploited by pyridine-negative mutants who benefit from increased iron acquisition without paying a cost. Experimental evolution has revealed that in spite of cheats exhibiting reduced relative fitness in an iron-limited medium, they can spread through a population of cooperators over a matter of days. And a recent study showed that the social selection may be similar, resulting in the pyridine mutants in the CF lung. And loss of function mutations occur in the pyridine synthesis genes, but not on the receptor gene, indicating that the loss of pyridine in vivo is not driven by disuse, but by loss due to cheating. And cheats can only exploit producers as long as they maintain a functional receptor. If loss were a result of disuse, mutations would be predicted to accumulate in both receptor and synthesis genes. However, with the notable exception of pyridine, we know very little in practice about the role of social evolution in shaping trait loss in the context of CF lung. And it could be tentatively suggested that virulence factors and secreted toxins may benefit bacteria other than the producer and hence may also be vulnerable to non-producing cheats. And if this was the case, the dominance of the loss due to disuse framework for explaining the loss of secreted traits in CF lung infections may open to uncertainty. Now, as far as the complexity of P. aeruginosa populations in the CF lung, it can be really complex and diverse in terms of phenotypes. Analysis have shown that there is extensive phenotypic heterogeneity within populations of P. aeruginosa in the CF lung beyond their colony and morphology. They show population diversity and many of the phenotypes to be altered during adaptation, including motility, virulence factor production, cytopore production, antibiotic resistance, oxytrophy, and hypermutability. Even though some of the population has acquired mutations affecting these phenotypes, these mutants coexist in patients alongside other genotypes and have not the most relevant concern of this diversity is antimicrobial susceptibility testing, which is typically carried out on either single isolates or two other colonies acting as a representative mucoid and non-mucoid colony morphotypes. But there's also diversity in the antimicrobial susceptibilities within the population, isolated from individual sputum sample. P. aeruginosa within the population diversity has been stressed to genomic analysis with reports that exist within patient genomic diversity between isolates and can be compared to the variation reported between sequential isolates. More studies have been done by analyzing larger numbers of isolates per sample to characterize the diversity present within populations. By sequencing the P. aeruginosa populations from a group of patients infected with the same strain, the study was able to show that the coexistence of divergent sublineages within each patient, predicting the likelihood of constant transmission between patients. The study showed the mutations in some genes are common. They're neither always present in all patients nor carried out by all affiliates of the P. aeruginosa population with each patient. And different areas of the lung tissue will vary along a range of environmental axes, like differences in mixtures of nutrients, concentrations of penetrating antibiotics, and other microorganisms or factors such as oxygen availability, potentially leading to variable selection for different ecotypes. And it's supported by the coexistence of distinct sublineages associated with different parts of the airways in patients. And then another study I found talked about the different regions of the lungs from chronically infected CF patients were analyzed to study the variations in clonally related isolates. The P. aeruginosa isolates occupying different regions of the lung had evolved independently, and they differed in phenotypic characteristics like nutritional requirements, antibiotic resistance, and virulence. And so there's a limited intermixing between these separate communities, which means that regional isolation mediated by spatial structure enhances the origin and the maintenance of the diversity. So diversity in CF lungs is adaptive and non-adaptive, and they enhance the origin and coexistence of the genotypes. Experimental tests are limited, and studies have shown that genetic diversity evolves in structured biofilm populations and the role of stressors in the CF lung and selecting for the evolution of diversity in P. aeruginosa biofilms and in populations growing in CF-like artificial sputum environment. The complexity is highlighted by the fact that diversity can happen within a biofilm population in a microcompartment in the wider CF airway environment where physically separate biofilm populations can be exposed to different environmental conditions, including variable antibiotic concentrations. Furthermore, as the lung tissue deteriorates over time during the CF disease progression, it's possible that the selective forces working on the pathogen population may vary, such that mutations beneficial earlier in the infection may be less favored. The effects of many selective forces likely to work in the lung still need to be tested, and these include other members of the microbial communities in the host immune system and phage. So to kind of wrap things up, because of genome sequencing, there's been a lot of progress in our understanding of how Aeruginosa adapts and evolves in chronic CF lung infections. More recently, this has extended to fine-scale analysis of evolutionary dynamics within infecting populations, revealing high levels of coexisting genetic and phenotypic diversity, including clinically important traits. The clinical consequences are not 100% understood, but given the extensive phenotypic diversity, there are clear implications for the false diagnoses based upon antimicrobial susceptibility testing using single pairs of isolates. There's limited effectiveness of antibiotics in these chronically infected patients. It is important that we improve our understanding of the evolution of bacterial populations during chronic infections in order to design better strategies for clinical intervention. And as time has gone on and we have advanced, we're able to figure out the structure and dynamics of bacterial populations during infections. And it's already helped us out so much. However, it is also important that we improve our understanding of the complexity of pathogen physiology and evolution. And one way to bridge the gap between in vivo operational data and an understanding of biological mechanisms is to use data from clinical samples to create hypotheses that can be tested subsequently using experimental evolution. And by doing this, we can make a huge impact in patients during the infection process. And that is it. And these are my references.